This is part 2 of chapter 10, and when we left off in part 1, I was mentioning the fact that these electrons that are lost from photosystems during photosynthesis get passed down electron transport chain. And I mentioned there's two different types of electron flow: non-cyclic, which is basically the default that we're really going to focus on here. But then we also have this possibility for what's called cyclic electron transport. And I'll show you the difference, the distinction between those two here. But I want to first focus on the non-cyclic electron transport. And here's what this looks like. So we're really dealing with two photosystems working together as a team. And these photosystems are called photosystem 1 and photosystem 2. Really, the only distinction between them is they have slightly different chlorophyll molecules in the reaction center that are optimized for absorbing slightly different wavelengths of light. But that's really the main distinction. But as you can see here, the way these work together is light energy that's being funneled into the center of photosystem 2, causing electrons to be lost from that reaction center chlorophyll, get passed down an electron transport chain, and then given to the chlorophyll in the reaction center of photosystem 1. And remember, this chlorophyll is also losing electrons constantly due to the light energy that's being funneled into the center of this photosystem. So if we don't want that chlorophyll molecule to just break down because its atoms are losing electrons, we need to replace those electrons. And so we're going to replace them with electrons that were lost from its partner photosystem, photosystem 2 over here. The electrons that are being lost from this photosystem 1 are then being passed to electron carrier NADP+, causing it to become reduced to NADPH. One other thing I wanted to point out here, really I guess two other things. One is you'll notice as electrons are moving down the electron transport chain between the photosystems, ATP is being generated. And we'll look in just a minute a little bit more detail at how that works. But this is one of the main products of this whole process is ATP. And then the other main product we already saw over here is that reduced electron carrier. So these are two energy-rich molecules that are being produced here. And we're just using the energy of sunlight to make that happen, right? It's the energy in these photons that's allowing us to produce this ATP and this NADPH. The other thing I wanted to mention is we do have an issue over here in photosystem 2. This chlorophyll in the reaction center is losing electrons and they need to be replaced somehow. And the way the plant does that is it will use water, so this is why we need water in the chloroplast. It will split the water molecule, take some of those electrons and donate those to the reaction center to replenish those electrons that were being lost. In the process of splitting the water, we end up with some hydrogen ions, but more importantly for us, we end up with oxygen, right? So this is just byproduct of photosynthesis, but it's obviously key for our cells to function, right, for cellular respiration. And so I realize this can be slightly confusing here. One point of confusion is that these photosystems are named photosystem 1 and photosystem 2, but photosystem 2 actually acts first, right, before photosystem 1. So I think as long as you can keep that in mind, you should be fine. But I do want you to have a good feel for this whole picture here, how this all fits together. I almost always ask a question on the exam. What is the flow of electrons during non-cyclic electron transport? And the answer would be, well, electrons are flowing from water to photosystem 2, down the electron transport chain to photosystem 1, from photosystem 1 to the electron carrier NADPH. So the reason this is non-cyclic, or sometimes you'll see it called linear electron transport, is these electrons do have a start point, right, and an end point. So the electrons are starting their journey here in water, and getting passed through this whole chain and ending up here in the electron carrier. So how is cyclic electron transport different? Well, cyclic electron transport is only going to be used in times of energy crisis, essentially. So if a plant cell is desperately low on its own ATP that it needs to power, other cellular processes, it can switch from the non-cyclic flow we were just talking about to cyclic in order to generate a little extra ATP. And I'm going to go back to the previous slide here just to be able to compare the two. But what's happening in cyclic electron flow is what's different is over here on the right with photosystem 1, the electrons that are being lost here are not going to be passed to NADPH. So in cyclic flow, we won't make any of that reduced electron carrier. Instead, those electrons will be passed back down the electron transport chain here, and they'll just keep cycling around, right? Hence the cyclic flow from photosystem 1 down the chain and back again. And remember, we talked about the fact that the more electrons that pass down this transport chain here, the more ATP we make. So that cyclic flow is going to produce extra ATP that we wouldn't be getting here in this non-cyclic flow that we see here. Right. So here's that cyclic flow, right? Photosystem 1, electrons lost, pass back down the chain, right back to the reaction center chlorophyll in photosystem 1. Now I want to focus in on that aspect of ATP production. So how does this work? How is that electron transport chain producing ATP? And technically this gets called photophosphorylation, but this is kind of a fancy word, just like we talked about, like oxidative phosphorylation during cellular respiration, which was just, we're phosphorylating ADP to make ATP in the presence of oxygen, right? In this case, it's photophosphorylation because we're phosphorylating ADP to make ATP utilizing photons of light. Okay. But the good news is if you are comfortable with the ATP production process in mitochondria during cellular respiration, you're not going to have a problem with this because it's basically a variation on the same theme. Okay, so here's a little bit more detailed view of that non-cyclic electron flow. Here's our two photosystems in pink, right? Photosystem 2 on the left, photosystem 1 on the right. We've got the electron transport chain in between the two photosystems. We've got these molecules here at the end of the chain that are passing electrons to the electron carrier. And you'll notice all these components are either embedded in or associated with that thylakoid membrane, right? We already talked about the fact that the photosystems were in the membrane, but this picture really gives you the nice context in terms of how everything is arranged. And so in this image, the blue down here would be the stroma, so the fluid inside the chloroplast, and the yellow up here would basically be the inside of those thylakoid discs, those kind of poker chip looking things that we find inside the chloroplast. Excuse me. And so again, the way this works, right? Our light is coming into the chloroplast, hitting these photosystems. All that energy is getting funneled into the reaction centers, and those reaction center chlorophylls are losing their electrons. And I'm going to focus on just what's going on between the photosystems here. So again, those electrons that are lost from photosystem 2 are being passed down this electron transport chain to photosystem 1. And just like we talked about in cellular respiration, as the electrons pass down this chain, a little bit of energy is being released with each passage. And some of that energy is being trapped and used to pump protons across a membrane. So same thing that we saw happening with the electron transport chain inside the mitochondria during cellular respiration. So we're pumping protons from the stroma into the thylakoids. So we're building up this big gradient of protons here. Because those protons all have positive like charges, they do not want to be next to each other, right? Like charges repel each other. Those protons want to get out of there, and the cell provides them a path to do so. And again, just like we saw in cellular respiration, in the form of ATP synthase, which is this enzyme that contains these channels for the protons to flow through, flow back across the membrane. And as protons pass through ATP synthase, they're causing this shape change down here in the catalytic subunit of the enzyme. And with each shape change, this enzyme is grabbing ADP, grabbing a phosphate, and bonding them together, so to form ATP. So the more electrons that flow down this chain, the more protons we pump, the more ATP we make. And just to complete the picture, don't forget we have down here, right, the electrons that were being lost from photosystem 1 getting passed to that electron carrier NAD plus to produce NADPH. So what we're seeing down here in the corner, again, are these two main products of the light reactions of photosynthesis. We're using light energy, these photons of light, to produce ATP and NADPH, both energy-rich molecules. Now, what do we do with ATP and then NADPH? Well, hopefully you remember the main point of photosynthesis was to produce sugar, right? And building a sugar is catabolic. That's a reaction that requires energy. It's got a positive delta G. So we're going to use these energy-rich molecules, ATP and NADPH, to provide the energy source to allow us to build those sugars. Yeah. So we've got our energy source. Another thing that we need if we're going to build sugars is carbon, right? You might remember carbohydrates are big carbon-containing molecules, and that's an issue for the plant. Where is it going to get all this carbon from? Of course, it could steal it from other molecules inside its cells, but then the plant is losing something, right? It's having to give up carbon from other molecules. So ingeniously, instead, what plants have figured out to do is they can just grab carbon from the air in the form of carbon dioxide, and it will actually, what's called fix that carbon, incorporate it into molecules inside the plant cells, and use that carbon from the CO2 as its carbon source for building those sugars. So this whole process would be the light independent part of photosynthesis that's happening out in the stroma, and the biochemical pathway that occurs that actually produces the sugars is called the Calvin cycle. Okay. And you're not going to be responsible for all the reactions or the intermediates of the Calvin cycle. But I do want you to know that it's divided into 3 sections. So the first part of the Calvin cycle is carbon fixation. So this is where we're actually grabbing CO2 out of the air and incorporating it into some existing molecules that are already part of the Calvin cycle. And this is the one reaction I do want you to know. So this reaction is so important. The enzyme that catalyzes this carbon fixation is the most abundant protein in the world on planet Earth. There's more of this enzyme, which is called rubisco, which stands for ribulose bisphosphate carboxylase oxygenase. It's a mouthful. You can see why it's just abbreviated as rubisco. But this is so important that there's more rubisco than any other protein in the world. And again, what Rubisco is doing is it's grabbing CO2 and it is bonding it to these molecules of RUBP that get split in half. So we end up with these little three carbon molecules essentially at the end of carbon fixation. Then we are going to actually produce the sugars that are going to be coming out of the Calvin cycle in the second part. And the sugars that we're making are not glucose initially. They're just these little 3-carbon sugars called G3Ps. Okay. So we're going to get a couple of G3Ps out of the Calvin cycle, and then the third and final part of the cycle is just regenerating that starting material. And you may notice if you look around the outside of the cycle here, we need a lot of energy for this to happen. So we're burning a lot of ATP, right? We spend 12 ATP over here. We spend another 6 over here during regeneration. We're using a bunch of those reduced electron carriers down here. So it's very energy intensive, but it's no problem for the plants, right? Because the plant was basically making this ATP and this NADPH essentially for free, right? Just using energy from sunlight. So no problem. The other thing I wanted to point out is even though we have a lot of these 3-carbon sugars in the Calvin cycle, we've got 12 of these G3Ps here, we're only going to pull 2 of them out for every turn of the cycle. So we're going to leave the other 10 in there and use those, as I was saying, to help regenerate our starting material. So bottom line, 3 parts to the Calvin cycle. I'd like you to be familiar with those. And I'd like you to be aware that the sugars that we're getting out, we're just taking out a couple of these G3P 3-carbon sugars. So there's those 3 parts: fixation, reduction, regeneration. And just to go back to kind of the broad overview and remind you where all this is happening. So here's our cartoon of the chloroplast. Here's our thylakoid, stroma down here. So we've got all the light reactions, the photosystems, right, the electron transport chain, all of that we talked about earlier was happening here in the thylakoid. The ATP and NADPH that get produced are actually being made in the stroma. If you go back to the cartoon of the photosystems, you'll see that they were actually being made on the stroma side of the membrane. And then all the enzymes that we need for the Calvin cycle are out here in the stroma, and we can use the ATP and the NADPH to power that Calvin cycle to make those sugars. You'll notice here they're referring to the ATP cycle and the NADPH cycle here between the light-dependent and the light-independent reactions, and that's just referring to the fact that the ATP that we make up here goes off to the Calvin cycle, loses a phosphate group and gets converted into ADP. The ADP then goes back up here and picks up another phosphate and gets converted to ATP. So we have this recycling. And same idea with the NADPH. So NADP plus picks up electrons, gets reduced to NADPH up here, goes to the Calvin cycle, drops off those electrons, gets oxidized, converted back to NADP plus, and then go back and pick up more electrons again. So a little recycling there. This was just to show you what the little 3-carbon sugars, the G3Ps that we get out of the Calvin cycle look like. You don't have to memorize that structure, but just so you're aware. And also to remind me to tell you that there is a video on Canvas that goes through all the steps of photosynthesis that I would recommend that you take a look at. It's not particularly long, but I think it's useful in helping to put all these parts together. All right, so just to finish out this chapter here, I had just a couple of loose ends to tie up. So I did want to point out, right, these photosynthetic organisms like plants would be considered autotrophs, meaning they can feed themselves, right? They're basically generating their own sugars, and can then use that sugar to power cellular respiration in their own cells. Right, so that's another important point. I hear this from students all the time. Oh, well, animal cells carry out cellular respiration, plant cells carry out photosynthesis. Well, that is true, but plant cells also carry out cellular respiration. They have mitochondria. They're going through that exact same process that animal cells are, breaking down the glucose, glycolysis, citric acid cycle. They just have the advantage of being able to make their own sugars, right? They can take those G3Ps that were made in photosynthesis and assemble those into glucose or other sugars. So anyway, autotrophs, self-feeders, this is in contrast to heterotrophs like ourselves, right? We have to eat other organisms, plants or animals, in order to get those sugars. So you've probably seen that distinction before. I also wanted to point out this important issue that can arise with photosynthesis, and this ties in with that enzyme rubisco. That's the enzyme that fixes carbon, right? Incorporates carbon from carbon dioxide into these molecules in the Calvin cycle. Well, if you remember the name of rubisco, it has both of these words in it: oxygenase, carboxylase. So the carboxylase part is referring to the fact that rubisco fixes carbon, but in the absence of carbon, it can also fix oxygen. And well, you might think, well, okay, why, how is it distinguished between the two? When would it do that? Well, really, this becomes an issue in very hot, dry environments. So imagine it's very hot and dry. The plant is going to close those pores on the leaf surface that we talked about in the very beginning of the chapter, the stomata, in order to try to preserve water and just prevent it from drying out and wilting. So we close the stomata, which we're also serving as our source of gas exchange, right? Allowing CO2 to get in and oxygen to get out. So once we close the stomata, we can't get new carbon dioxide in, so the plant cells are going to use up the carbon dioxide that was in the cells already, and then it's run out, and the oxygen that's being produced as a byproduct of photosynthesis can't get out of the leaf cells anymore. And so what will happen is, in this situation, if there's a lot of oxygen and very little carbon dioxide, that rubisco enzyme will start fixing oxygen instead of CO2 in the Calvin cycle. And bottom line, this is not good for the plant. So it's going to produce these toxic compounds that are not going to be healthy for long-term survival of the plant. So a plant really can't deal with that situation long term. So what I wanted to point out is that there have been several strategies that plants have evolved to get around this issue. So one is called photorespiration. We're not going to go into the details on any of these, but I did just want you to be aware that this is a problem and just have a basic idea of what these strategies were. So in photorespiration, the plant is going to take those toxic intermediates that were produced due to the plant fixing oxygen and basically biochemically convert them back into non-toxic, usable molecules that will allow the cell to complete the Calvin cycle. In C4 plants, these plants get around this issue by moving carbon fixation into a physically different location in the cell where there's not going to be as high a concentration of oxygen. CAM plants take yet another strategy. They will basically only fix carbon, only run that part of the Calvin cycle during the cooler parts of the day when the stomata could be open. Okay? So that we wouldn't have that problem of oxygen building up. All right, and just last slide here, just to end things. I did just want to point out that photosynthesis, even though it's amazing and works great and basically powers all life on Earth, is really not particularly efficient. So if we think of the energy, that was coming in and hitting these plants in those photons of light initially, we're really only capturing 5% or so of that energy in those stored sugars. So a lot of it's being lost just due to various inefficiencies, but it's like I said, it's enough to power all of life on Earth. So we're not too worried about those inefficiencies. All right, that is the end of Chapter 10 in our overview of photosynthesis.